All right, ladies and gentlemen, so welcome back. We're going to continue our discussion on classification of organisms by really talking about phylogeny today. So, phylogeny is just this description of evolutionary history among either a single species, tracing it back, or looking at a group of different organisms and what's the connection to each other based on evolutionary history. So, phylogeny, there really is a connection between that classification scheme of things like order, genus, species to their phylogeny. And we pull all of this information together and talk about phylogenetic trees. So that's what you're looking at here in the picture. This is an example of a phylogenetic tree. It's a hypothesis about the evolutionary history and the branching divergence points that would be reflective of the organisms within your particular study. And this particular one happens to also layer on top of that the different classification groups. So the orders, the families, the genus, and the species is what you're seeing in this particular example. So that those classification fields do end up sort of nesting within each other as inclusive groups up to that bigger order, in this case, like the carnivores. So we're layering on top of phylogeny and Linnaean classification in this particular example. That being said, those phylogenies can definitely differ from each other compared to their sort of traditional classification schemes when we start recognizing other pieces of information. So let's just talk about how these phylogenetic trees get built. All right, whether we're utilizing just traditional classification information or we're taking into account genetic information or fossil records or whatever the case might be, the basic building of the trees occurs the same way. And the way that you read the trees occurs in more or less the same way. So when we look at phylogenetic trees, they represent, again, these hypotheses about their evolutionary relationships between a set of organisms that we have chosen to study. Every time that you see a branch point, that represents the divergence of the common ancestor into two or more distinct groups. All right. And then within those sets of groups where that branching point occurs, we can talk about these things called sister taxa. So they're groups that share an immediate common ancestor. So if you come back up here and take a look at some examples, here's a sister taxa, right? So they share this common ancestor. It's immediate to the branch of both of these two species. If we look at D, E, and F, they are all also sister taxa because they all come back to the same branch point where instead of it branching into two, it actually branched off into three. We can talk about rooted trees. These include a branch to represent the last common ancestor for all of the taxas in the tree. So this would be, in this particular example, a rooted tree back to the common ancestor to everybody in this particular example. And then your basal taxon, or you may come across this term as an outgroup, is the one that would have diverged first compared to everybody else in the tree. So it's sort of the oldest organism known in that tree and would thus share the least amount of common characteristics with everybody else in that particular group. This term polytomy comes up every now and again, so that's where we'd get this example with the D, E, and F, where that branch actually goes into more than two groups, so rather than just being a typical sort of fork in the road where you either went one direction or another, it was a more widespread change to actually create multiple offspring or multiple offshoots, if you will, as opposed to just a single one. So here's the thing. There are definite benefits to these phylogenetic trees, and then like everything else, they have their own challenges, right? So we can learn certain things from them and not others. So they definitely show patterns of descent, for sure. They may or may not be particularly good at showing phenotypic similarities between organisms, depending on how the tree is built. They don't necessarily indicate where the species evolved or how much change has occurred in that lineage. We'd have to adjust the way that we build these trees, and there are some options out there we'll take a look at that can show a little bit more of that. And these also assume that the taxons evolved from those next to them. All right. We have to be careful with that assumption because that's not necessarily always the case either. You've got to trace back where those branches are coming from. So there are a bunch of options when we build these trees. A typical phylogram here will actually show the number of changes in DNA that take place in a particular sequence that we're studying and using as a way to build out this evolutionary relationship. So if we look, something like the Drosophila clearly has a lot of changes and thus makes sense that it would have branched off much further back than when we look at some of the organisms that are more closely related here, like the human, the rat, or the mouse. Ultrametric trees, these now layer on top of them chronological time. So this one's happening to show some of that geologic history here as it relates to when those branch points actually occurred. Okay, so it puts into context some of that geologic time. Usually we're utilizing ultrametric trees when we're taking data specifically from fossil records. Your phylogenies can definitely provide important information about similar characteristics in closely related ones, to the point of actually being able to sort of identify unknown information. So this is kind of an interesting example where forensic ecologists were actually using phylogeny and known characteristics to identify illegal whale meat. So poachers would go out, hunt these endangered species, try to sell the meat at auction or in marketplaces. Oftentimes we come across places like this in like Asia and Africa and things like that. And so these forensics ecologists were utilizing characteristics, probably genetic ones or protein sequences from the meat that they were getting out of the marketplaces and tying it back to whether or not they corresponded to a particularly known species. So these were just unknown samples that they were showing, and based on that genetic information, we're able to make assumptions about their closest relatives, which is pretty cool. So when we're going to build out this information, we're typically inferring these phylogenies or these evolutionary histories from three major sources of data. There are morphologies, so physical characteristics that we can see, whether it's because we're dealing with living organisms, or this is typically the one we rely on the most heavily if we're looking at fossilized organisms. And then the genes and their biochemistry are actually much more common when we talk about living organisms, especially more recently. We're focusing a lot on their DNA sequences, and then how do they express them. So usually it's some interplay of DNA sequences, messenger RNA sequences, and then their actual protein sequences as well. So when we look at morphological and molecular homologies, right, that's what we're using here, those homologs, those sets of characteristics that are similar between organisms. Do they have them or do they not? So phenotypic and genetic similarities are due to common ancestry. We know that. So the more genetic structure, the more protein structure that organisms share, the more closely related they must be. So the more differences we see in those sorts of sequences, then the more likely they diverged further back in time. It's sort of like putting fossils into that striated layer, right? The further back you go, the more layers are above them. The closer they are to each other, the more likely they are to be closer in time and potentially more likely to be similar to each other. So when we're looking at the larger number of differences, in this case, between their morphologies or their DNA sequences or their protein sequences, the more likely their divergence must have happened much further back in history than if they share most of that sort of thing in common. So we look at these ideas of constructing these phylogenetic trees based on similarities. Now, something we have to be careful of is the difference between, especially when we look at morphological data, physical data of characteristics, we've got to be careful between the ideas of homology versus analogy, right, an analogy. So a homologous structure is one that truly comes from a shared common ancestry. So we're talking about similar bone structure, muscle structure, neural structure, the way that those structures develop from embryo to adult, as opposed to analogous structures, which are really a matter of convergent evolution. And so we're looking at organisms converging or coming together on similar traits because they fit a similar niche in their environment. So we look at this. This is homology, right? So we shared a common ancestor, human, mouse, and wing, or bat wing, and we've got similar bone structure, although the bone length might be a little different, thickness might be a little different, the basic orientation is the same. The nerves, the blood vessels, the muscles that all connect to those bones also are of a similar origin, not only in terms of the way that they're structured as an adult, but also the way that they develop from embryo to adult. That's true divergent evolution. That's true Darwinian evolution. We look at convergent evolution, we're now looking at these similar adaptations in different species that really don't share a common ancestor without going really far back into history. But they share a similar niche in their environment. So this is convergent evolution, the bat wing, the butterfly wing, and the bird wing. So the way that they're structured, the way that they develop in embryo to adult is all different, but they have all converged on this wing structure because they all fly, all right? So we've got to be careful when we're talking about using physical characteristics. Are they truly a result of divergent evolution, in which case we share common ancestry, or are they a result of these convergent evolutionary events where these organisms are coming together on similar structures by chance because they happen to fit a similar role in their environment? Molecular homologies are why we kind of tend to lean towards this now. We're getting much better at doing DNA sequencing, molecular sequencing, protein sequencing. It's getting faster, it's getting cheaper, it's getting more accurate. And so at least with living organisms, this is typically where a lot of scientists are going because we know for sure that this is a result of divergent evolution and not because we've happened to converge on something, or at least we're much more confident in that analysis. So we also use all sorts of computer programs, mathematical tools. We'll play around with some of this stuff later in the year, but we're looking at molecular systematics using the DNA and other molecular data to determine evolutionary relationships. So the more of that DNA sequence or the more of that protein sequence that is identical in different organisms' genomes at a similar location for a similar trait, the more confident we are that they are closely related, at least with respect to that particular characteristic, as opposed to seeing large numbers of differences. The more different they are for that particular characteristic, the more likely they diverged further back in evolutionary history as a result of that.